All right, we'd like to start with our next set of speakers. I'm again honored to introduce Dr. Sandra Weller, who was a graduate student with Dr. Temin. For many years, she was perhaps most well known as the scientist working in a lab on a normal day in 1975, whenever Howard Temin received the news that he was awarded the Nobel. She has since become an internationally recognized molecular virologist. She is at the University of Connecticut, and she has been a molecular virologist for more than 4 decades, studying the elucidation of replication strategies of herpes simplex virus. She has also developed innovative antiviral approaches and uncovered evolutionary connections linking herpes viruses, bacteriophages, and ancient genetic elements. Dr. Weller has served for 17 years as the chair of the Department of Molecular Biology and Biophysics at the University of Connecticut School of Medicine, and she also holds the title of Board of Trustees Distinguished Professor. In 2022, she was elected to the American Academy of Arts and Sciences. Let's welcome Dr. Weller.
Thank you very much. This is working? This is working? Okay. It is such a pleasure to be here and to be back in Madison, and I'm just so grateful for my time here and for you inviting me back. So there is a reason for my all the world's a phage. Does anyone know who coined that term? Yes, absolutely. Dick knew. And you'll see why I showed that. Okay, so this was quite a day. I put it up here as well. I just have one correction to what Rayla told you, which is that I was not actually doing an experiment that day. I went in there because of all the commotion and it was so exciting. And there was a TV camera, and so that's kind of a fake. But just that's the only fake of my entire scientific career. I haven't faked anything else. Okay, so what I want to talk about, and it's related to my own career, is what we've been hearing before, but this idea of intuition, perseverance, backed up by data, has driven my career as well. And I think that there are at least 2 kinds of scientists, some that are incredibly data-driven, and some people that are maybe more intuitive. And I feel like I learned that it was okay to be intuitive from Howard, because he was. Obviously, he backed it up with data, but he was intuitive. So the journey of when he first had the provirus hypothesis, as we heard from Rayla, was in 1960. It was published in 64. Between 1964 and 1970 was a tough time for Howard, because he'd go to meetings and people would think he was crazy. And that's hard, when all your colleagues laugh at you and think you're crazy. So the definitive proof, of course, came, and the rest is history. So my title is sort of Good Cooking and Science Takes Time. And I'm going to tell 2 stories today. One is a project about my thesis project, which took 21 years. And I'll show you, I was not here for 21 years, but the project to come to fruition was 21 years. And then I'll tell a story about my career since becoming a professor at UConn. So in my first year of graduate school, I found this thing that was strange. I found that some subgroups of avian leukosis viruses were killing cells. And I found that there were subgroups B, D, and F. I told Howard that these viruses were killing cells, and he told me that retroviruses don't kill cells. In my memory, I've added the line, first-year graduate students kill cells. So for a long time, I don't think he quite believed me, but I did finally convince him that these 3 subgroups of viruses were killing cells. They didn't kill them off entirely, and the cells grew back, and they still transformed, or whatever they were going to go on to do, because retroviruses in general don't kill cells. But I found that the cells that were dead, I separated dead and live cells, and they contained a lot of unintegrated viral DNA. And the only way you can get unintegrated viral DNA is by superinfection. You see, a single cell may be infected by many viruses. And we knew that cell killing was caused by the envelope, but we didn't know why. It took a long time. It was left a mystery. We didn't know why. And 16 years later, we found out why. And I was organizing a meeting, a FASEB meeting on virus assembly, and I had invited John Young. John Young had worked with the Harold Varmus and had cloned the first A subgroup virus receptor. And I saw Harold at that meeting, and I said, you got to do the B or the D. But 16 years after I published what I published, he actually talked about, I'm supposed to do it this way. Is that working still? He talked about cloning the B subgroup receptor, and it was a member of the TNF family subgroup of surface proteins, and on binding a cellular viral envelope, either from the B or the D subgroup, he could induce apoptosis. And this was really the first time that anybody showed that a signal from the outside could induce apoptosis. So 16 years later, I figured out that I had discovered apoptosis, which wasn't really true. It was known, but I felt like I could contributed something to it, and it was really exciting. Okay, so story 2 is when I moved to herpes viruses, and I became a postdoctoral fellow with Dr. Priscilla Schaefer at the Dana-Farber Cancer Institute at Harvard. And you may wonder why I left retrovirology. And it's already been alluded to, but I really didn't want to keep working on a virus that only caused cancer in chickens. And so I picked viruses that actually cause a whole lot of diseases, as many people in this audience know. And 6 months later, Bob Gallo comes to the Dana-Farber and gives a seminar on HTLV1, and I'm like, there is a human retrovirus? Really? I mean, for the whole 10 years of Nixon's 10 years of the fight against cancer, we were trying to find a human retrovirus. It just took a little while. So when I started in Priscilla's lab, that we knew a little bit. I mean, this was in 79, 80, and we knew that we didn't know much. Only one. So we knew that it had a large genome, and it had the size of the genome was actually determined in 1971 as being close to 150 kilobases. We knew one gene was mapped. DNA polymerase was mapped in the middle of the unique long arm of the herpes simplex virus genome. We knew that Priscilla Schaefer, when she was a postdoc, she and several of her colleagues in Joe Melnick's lab at Baylor had isolated a lot of temperature-sensitive mutants, and some of them were DNA negative. So we thought, okay, if you have a DNA negative virus, it's probably maybe. I was interested in DNA replication, and I wanted to learn more about some of the genes that carry out DNA replication. And so cloning had just come on the scene. We got access to a R1 library that was made by Alan Golden and Roz Sandry Golden. So that became available. The physical mapping of the polymerase gene was done in Priscilla's lab, or in collaboration with Neil Wilkie and Pierre Chartrand and Clyde Crumpacker. And one of the origins had been mapped, and it had been mapped as a repeated gene on the unique short arm. So there were 2 copies of ORES. But that's what we knew when I started. And the work that I did as a postdoctoral fellow was to first physically map the gene for the single-strand DNA binding protein that was known to be encoded by herpes, and it was known to be essential because TS mutants were DNA negative. And I started working on ICP8, and I am still working on ICP8. In fact, at the end, I'm going to show you a structure of ICP8 bound to single-strand DNA that was done in my lab. So I'm very proud of that. In mapping the ICP8 gene, we found that the region between DNA polymerase and the ICP8 gene could not be cloned. No one had been able to get clones. All the clones had deletions. And it was kind of a mystery of what's going on. Why can't we clone this gene? And so I spent the next 2 years after mapping ICP8 and some other genes as well, trying to figure out what was going on. And what we found was that the region between ICP8 and the middle of two divergently transcribed genes was the origin of replication, which we now call ORAL because it was in the unique long arm. And what we found, and I was working with Antonella Spadaro, a postdoc in Alan Maxim's lab, and what we found is that this region was a 144 base pair perfect palindrome, 72 bases on a side. And I remember being in the lab one Saturday in Alan's lab, and I was reading off the sequence, and then I realized I was reading the same sequence on the other strand, and what? So this is how that happened. And so those are my two big projects, and it got me a job at UConn. Just a disclosure, I have a co-founder of a small antiviral therapy company. So during the first decade of my tenure at UConn, we mapped a lot of other genes. So we mapped we'd already mapped the ICP8 gene, we mapped the helicase primase, we mapped and discovered which 3 subunits were in the helicase primase. We studied the UL9 origin binding protein. And so we did a lot of genetics. I think I made the first blue herpes. So we had integrated beta-gal into something that we could integrate into the, and maybe I can thank Bill Resnikoff for teaching me about that, or Charlie Yanofsky, but okay. So then we turned our attention to to the mechanism of replication. So this is not a retrovirus. This is a large DNA virus, 150 kilobases, and its mechanism of replication, I would say, is still not completely clear, but we think we've made some inroads. So what we do know is that the virus, when it infects, inserts a double-strand DNA genome into the nucleus, which then replicates through some kind of very complex mechanism to make X and Y junctions. These don't enter a pulse field gel. If you look at them on EM, you see them in this very complex form. And yet, what we do know is that this mess has to get resolved into a concatemer, which then can be packaged into a capsid. And this is similar to a lot of other bacteriophage and a lot of other viruses from all domains of life, and even from a genetic element even before the evolution of cells, which is the ability to replicate through concatemers. And I'm going to talk about what we think is the mechanism by which it does this. So what's responsible for this complexity? Why did it make forks and X junctions that you can't, what's going on? And it's been known since the early 70s that the HSV genome contains nicks and gaps. And replication through a nick or a gap would be expected to activate host DNA damage. Even just the presence of a nick and of a single-strand region next to a double-strand region would activate host DDR pathways. And what we realized is that the viral genome entering the cell does activate several DNA damage or pathways. And so the major DNA pathways we would be looking at would be DNA PK, in non-homologous end joining, ATM. It's not working again. ATM is part of the homologous recombination system, and ATR ATRIP, which is normally seen at a stalled replication fork, and it causes checkpoint degrees. So those are the 3 major DNA damage sensing kinases that cells have. And we found first that the virus does activate DNA PK because it has ends and it has these gaps. It does activate ATM. You get ATM phosphorylation. You don't see ATR activation. And the reason for all these things is that HSV inactivates these pathways as soon as it activates them. So the fact of a damaged DNA going into a nucleus induces damage. However, that's then countered by the virus inactivating those pathways. And as was mentioned just before by Michael, this is part of a host, virus cell DNA pathway that, battle that's been going on for eons. So the virus activates these things, but then it inactivates them. And so the challenges for HSV would be to deal with this nicked and gapped genome in a way that can produce viral genomes that can then be packaged into virus. So those are the challenges, because you have a nicked and gapped genome, you have to worry about triggering host DNA damage responses, and you have to produce a linear concatemer. So in the meantime, when we started looking at these activations, and when we first saw activation of DNA damage responses, we thought, oh, the virus must have evolved to use those pathways to replicate its genome. The first, maybe Occam's razor, was to say those are going to be used by the virus. But we and others found that HSV actually has evolved to inactivate the pathways. So ICP0, which is a viral immediate early protein that's a ubiquitin ligase, can induce degradation of both DNA PK and several components of the homologous recombination pathway system. So it inactivates non-homologous end joining and HR, which are the two most likely pathways to cause circularization if that's what had happened to this virus. So the virus goes in as a linear, and we'll talk about circularization in a minute. HSV also inhibits non-homologous end joining through its nuclease, and I'll be talking about the alkaline nuclease in a minute. And the HSV repair replication proteins, elegant work done by Kareem Moni in my lab when he was a student, and he's now at the Mayo Clinic. And if you haven't yet, you should invite him down to give a seminar, because he has a really nice story. He showed that 4 replication proteins of herpes simplex can inactivate ATR can prevent ATR activation, and it's by sitting at the replication fork and not allowing ATR to notice that single-strand to double-strand transition. You would have it a nick. We also showed that, or we didn't show this actually, the HSV replication machinery is inherently ill-behaved. And when I say that, it was a beautiful experiment done by Bernhard Matz in Germany, who showed that SV40 norm is a circular molecule, very well behaved. It replicates by bidirectional replication, makes 2 circles, they distangle, you make new DNA circles. But if you replicate the SV40 genome in an HSV-infected cell, in the presence of SV40 T antigen, you get those same complex junctions, X and Y junctions. You get a lot of recombination. It looks like herpes replication. And by that, we all thought at the time, and I think it's turned out to be true, that the DNA replication machinery itself is inherently recombinogenic in some way. So the prevailing model for DNA replication in herpes simplex virus was that the viral genome circularizes upon entering the genome, entering the nucleus. You get bidirectional replication from one of the origins, and then magically you get a nick, and you then do rolling circle replication. And this is what is still in all the textbooks of how herpes simplex virus replicates. But there's a lot of evidence that actually doesn't support this model, and one of them is that if you look for actual circles in an HSV-infected cells, you don't see them. And this was a wonderful experiment done by my colleague. He was a postdoc in the same lab I was in, and he's now at Pitt, Neil DeLuca. And he showed that in an infection in which ICP0 is present, and remember ICP0 is the ubiquitin ligase that induces degradation of non-homologous end joining, in a cell that has ICP0, you don't see any circles. So indeed, non-homologous end joining has been inhibited. So in 2003, based on evidence, but some degree of kind of some intuition, I feel like that I kind of stubbornly stuck with this model for a while, but we suggested that HSV replication utilizes some kind of a recombination-dependent mechanism for replication. And this work was definitely inspired. I fell in love with viruses when I was 16 years old. I lived in Pasadena, California. I actually was able to work at Caltech. in a T4 lab, and I fell in love with viruses. So I have always been very interested in how the bacteriophage do things. And what I realized, and especially after meeting Gisela and Mosek, was that viruses like T4, Lambda, and T7 utilize single-strand annealing for doing replication and recombination. And one of the seminal observations that Gisela made, was that that you can prime DNA synthesis on a lagging strand by annealing a fragment to that strand. And that might seem trivial right now, but you'll see how that has led to the model that we have been pursuing. So I just wrote what I hope is my last renewal of an R01 that I've had for 45 years on genetics of HSV DNA replication. And I spent months going through the evolutionary data, looking at phage, looking at viruses that have DNA genomes that replicate through concatemers from all walks of life, all domains of life, even predating cellular evolution. And what we realized, so I've spent the last 22 years trying to get evidence for this, right? Going crazy. In 2014, I was asked to write a review that I could write with one of Don Kort's postdocs, Jim Sawicki. And so we wrote a paper showing the similarities between Lambda and HSV, and that has continued. So it turns out that viruses from all domains of life, as I just said, that do concatemer formation, they all encode a conserved two-component recombination mechanism, two-subunit enzyme that can do recombination. And the best characterized, of course, is lambda red alpha beta, which I learned about when I was a student here from Fred Blattner. And we'll talk about this mechanism. And all viruses from protozoa, bacteria, mammalian cells make a two-subunit complex. And in the herpes system, this is carried out by ICP8 and the enzyme alkyl nuclease. So using a 5' nuclease that generates single-strand ends and an annealing protein, you can do a lot of something called single-strand annealing. And I'm not going to go through all the properties of these proteins, but it turns out ICP8, in addition to being a classical SSB, which works in replication to stimulate other replication proteins, it also can anneal. It's a very potent annealase. And in fact, all the phage and all the viruses make a potent annealase. UL12 is an alkyl nuclease, which is a 5' nuclease. And as I will show you, together, these two enzymes can cause wreak a lot of havoc. We recently solved the structure the first structure of UL12, which is the HSV version. The SOCs had already been done, but we did the first one of these. So the data that's important, and I'm just going to show 2 slides, is that we showed that these 2 proteins, if you purify them together, could carry out strand exchange over long distances, as much as 7 kb. We also showed, using cell-based assays, looking at the activation of either homologous recombination, single-strand annealing, or the other one, that HSV1, UL12, and ICP8 could stimulate strand annealing, and it actually inhibited HR and non-homologous end joining. So our model, which is what I'm going to end with, is that HSV can take a single-strand fragment, and the nuclease can degrade the one strand, leaving a bottom strand that is coded by SSB, which can hybridize, and I'm done. I'm supposed to be done. I'm going to go really, really fast. Can hybridize at a lagging strand template. And our model is that repeated rounds of annealing at a lagging strand template, viral DNA polymerase doing strand displacement synthesis, creating new forks, generates a very complex condition which actually in the lambda system can lead to assimilation on a lagging strand of an entire lambda genome of 30 kb. So you can assimilate a lot of DNA this way. So our model is that you get repeated rounds of fork generation. You need a resolvase from that very branch structure to be able to package the DNA. And the next chapter, which I'll just mention briefly, is I was lucky enough 9 years ago to marry this man up here. He's a medicinal chemist, and so we have been collaborating on drug discovery. So we have a lot of really amazing chemotypes of drugs that can inhibit UL12 in particular, and they are very broad spectrum because that enzyme is very well conserved among all the herpes viruses. And we have recently solved crystal structures of UL12 and ICP8 with single-strand DNA. So these are the takeaways. The evolution story has been fascinating to me, and really the fact that genetic elements that have an exo and a single-strand binding protein can do this kind of reaction even before cells evolved tells me the importance of this evolution. So I agree with Michael that evolution is really important. And I had a semi-retirement party last year that was 40 years of virology. And again, I'm going to end with good cooking takes time, and my lab manager made this cake all 4 decades of my career in herpes, and what we worked on in each case, and you can see the bottom right panel. This is all made in sugar. You could eat this entire thing. Is a little pill bottle with drugs. So that takes us through the entire thing. This is my lab now, and the lab with my husband, Dennis Wright. We had a going away party last week for someone who had to leave because of our new president. And that's it. Thank you very much. Sorry for going over time. You probably don't have time for any questions.
Time for one question.
Hey, Sandy Covey. A great talk. Hi. I just had a question. So when you have that SSB-coded DNA, does UL52 primase activity ever play a role in allowing for replication? So if you have a UL52 knockout or inhibited it, does it still replicate?
So it's a complicated story because herpes genetics is always pretty complicated. But UL52 mutants are actually dead, but they're only dead for the first step. We think that during origin-dependent DNA synthesis, you need the primase. But we think that the priming by DNA that can happen subsequently, we think, is responsible for most of the DNA replication. And we haven't proven that exactly, but this is our model.
Cool. That makes a lot of sense. So, in my postdoc, I studied Primpol, which has UL52 herpes virus elements in the human genome for that specialized polymerase.
The Pol alpha primase?
That's not Pol for primase. It's a newer one that was discovered called Primpol. It has DNA-dependent repriming activity. And the DNA primase in that case is actually UL52, or has UL52 elements integrated.
That's very interesting. Let's talk about that.
So I think they might be similar in ways.
That's really cool. Okay. They're going to drag me off the stage with a fork in a minute.
Let's thank Dr. Weller.